Hello, students. How are you all? It's my pleasure again to talk to you, and especially for class 12 students who have taken PCB and now they are going to appear in the final exams on the 27th of this month. So, I am sending notes, question answers, along with my teaching to some of my students, and I'm also trying to make some videos on important topics which may help others also. So this video is on Lac Operon in E. coli, which is very important and questions generally come from this topic. What is Lac Operon and how do you represent it symmetrically like this? So first of all, you must have understood the chapter and you have to revise it, like what is Lac Operon and how does it function. So operon, as you all know, is a cluster of genes, and this is actually the idea how bacteria or E. coli utilize the sources for their energy in the best possible manner. Because lactose, glucose, all are there. Now they prefer glucose more than lactose as the source of energy. Now, if lactose is there, so it has to be hydrolyzed, it has to be metabolized to break down into galactose and glucose, which will be the best source of energy. So they operate in on and off mode. When lactose is there, it will be on because it has to be broken down, and off mode will be there when glucose is already there. So this process of breaking down or hydrolysis is not required. So these are the conditions for operating, and this operon is actually doing this. And how it does, because it is a cluster of genes, so anything to happen, you have to coordinate between a group of genes. If it is a group of genes, so every gene has got its specific function to carry out this function, to carry out this process. The goal is to hydrolyze lactose into glucose so that the bacteria gets the best possible source for its energy. For this, you have to understand the structure of the operon. As I said, it is a cluster of genes. So there are two types of genes you have studied also already. So one type is the regulatory genes, and the other is structural genes. The both groups together carry out this function. How, that we'll see now. So these regulatory genes are gene I, that is inhibitory gene. It will stop the transcription because somewhere this transcription has to be stopped also. Then it will be an off mode. And then the second gene is, it has to be started again. So whenever there is a need, so there will be a gene for this, which will is gene P, that is promoter. So it releases RNA polymerase, which will help in starting the process of transcription. And the third regulatory gene is gene O, operator, which is there in between the promoter and the other genes, so that when glucose is there, it will be in off mode, and when lactose is there, it will be in on mode. So these are the regulatory genes, and now we come to the structural genes. Structural genes are gene Z, which will help in the hydrolysis, actual hydrolysis of lactose into glucose and galactose. So this is gene Z, and this will be helped by the other genes like your gene Y, Gene Y will increase the permeability, semi-permeability of the cell so that maximum lactose is pumped into the cell. So this is the role of the gene Y, and then the other gene is gene A, the gene A, which will control the unwanted elements, and it will control the detoxification also. It will help in detoxification. So like this, the process is carried on, and then finally, what will happen? This mRNA, mRNA or this polymerase, RNA polymerase will release one single mRNA, which will be translated into 3 enzymes, and these enzymes will finally bring out the lactose metabolism in E. coli or in bacteria. So this you understood the roles of different genes in the Lac Operon, regulatory genes and structure genes. Okay, so I end this video here only and hope that this will help you in your remembrance and in your preparation for the exams. Thank you. God bless you all. Wish you all the best.